and the next presentation is also on circadian clocks, and it's going to be Kelsey Teeple from Purdue University, and her presentation is entitled Disruption of Circadian Clocks in the Prepartum Dry Period Negatively Impacted Mammary Development. So Kelsey, you can share your screen and get started.
Thank you for the introduction, Dr. Borman. Let me pull this guy up. As previously mentioned, I will be presenting on the disruption of circadian clocks in the prepartum dry period negatively impacted mammary gland development. To begin, I would like to start off with some background information. The dry period is a 45 to 60 day management strategy used in order to maximize milk yield. During this time, major tissue construction occurs in the mammary gland, such as high levels of programmed cell death of mammary epithelial cells in the early dry period, followed by high levels of mammary cell proliferation in the late dry period. Mammary epithelial cell number determines the milk production capacity of the cow. Environmental and management factors affect milk production and are related to dry period mammary epithelial cell proliferation. However, it is not fully understood how the cow integrates environmental and management information to affect lactation. One way that the cow can integrate environmental factors is by the circadian timing system. Circadian clocks generate circadian rhythms to coordinate physiological and behavioral processes to a 24-hour period. Clocks function to synchronize processes to be predictive of regularly occurring environmental cues, such as light and dark cycles. Studies in rodent and cell culture models suggest that the circadian timing system regulates mammary development. Circadian rhythms regulate homeostasis, but its role in lactation is not understood. In the image below, this depicts how the cow can integrate the light signal starting at her eye level and is then coordinated into her hypothalamus, where the master clock is located. From here, the master clock sends neurological and hormonal signals throughout the rest of the body into the peripheral clocks to align them to the cycles. And there is one in the mammary gland, and that's what we're specifically interested in. We hypothesize that disruption of the circadian timing system during the dry period negatively affects mammary development. Our objective was to knock out the function of circadian timing system in exposing cattle at 5 weeks prepartum and to a chronic jet lag model and measuring the effect on mammary development at 3 weeks before expected calving and 3 weeks postpartum, as well as milk yield to 60 days postpartum. In the image to the right, we have our two treatments, the control group and the phase shifted group. The control group had a chronic exposure to 16 hours of light and 8 hours of dark. The phase shifted group had the same amount of light and dark cycle. However, every 3 days, their light time was shifted forward 6 hours. For our study design, 12 multi-Paris Holstein cattle were split into two groups, 6 in the control and 6 in the phase shifted. At 35 days before expected calving, they started their light treatments and were on these light treatments until about 3 days before expected calving. From here until 60 days in milking, they all had the control lighting. At 21 days before expected calving and 21 days postpartum, mammary gland biopsies were collected. And from here, the biopsy tissue was stained with hematoxylin and eosin, as well as Ki-67 immunostained. H&E was used to measure the lumen to alveolar area. In the image below, we can see an H&E section of the mammary gland where the alveoli, or the milk secreting unit of the mammary gland, is stained. We first measured the lumen, or where the milk is collected into, and then we measured the total alveolar area. For Ki-67, we use this to count the proliferating cells. We started with counting the proliferating mammary epithelial cells, non-proliferating mammary epithelial cells, proliferating stroma, and non-proliferating stroma to form a proliferation index. Now we're going to start transitioning into our results. First up is the proliferation index for the mammary epithelial cells on the left, and then percent stromal cell proliferation on the right. The green represents our control group, and the gold yellow represents our phase shifted. The solid colors are our before expected calving time point, and then the stripes are our postpartum time point. Here, the treatment and time point had an overall effect on mammary epithelial cell proliferation. For stromal cells, only time point had an overall effect. The different letters indicate a difference of p less than 0.05. In the graph on the left, we can clearly see that the before expected calving phase-shifted group had a significantly decreased level of proliferation when compared to the control group. For the lumen to alveolar area, time point had an overall effect, as well as treatment had an overall effect. When comparing the treatment or control group compared to the phase-shifted group combined, we can see that the phase-shifted group had a significantly lower amount of lumen to alveolar ratio when compared to the control group. Finally, our last measurement was milk yield. We collected milk daily until 60 days postpartum. There is a significant difference in the milk yield. We can see that the control group had a significantly higher level of milk production compared to the phase-shifted group. In conclusion, phase-shifting the light-dark cycle decreased mammary epithelial proliferation in lumen to alveolar area, which indicates that circadian disruption in the prepartum dry period negatively affected mammary development. Lower mammary development was related to lower milk yield in the postpartum. The dry period is a critical time for mammary development, and maintaining a consistent management schedule could minimize potential milk loss. Thank you to the USDA grant that funded this research, and to all the staff of the Animal Science Research and Education Center in Purdue, and then to all the undergrad students and faculty members who helped with this project. Thank you.
Thank you, Kelsey. And we are a little bit ahead of time of the schedule. So if someone has any questions, they can submit them. I do have a question, it's really hard to come up with them as we are going so quickly through all of your information. You showed a lot of information, but for your lumen to alveolar ratio, there was a lot of variation on the postpartum cows, the control cows postpartum. Do you have any ideas on why there'd be so much variation in that group, or is that a measurement that's pretty hard to do?
I am not too sure for that. I would expect that for the variation, it represents the degree of differentiation. If some cows happen to, be a little more sick, or if they had a little bit delayed onset of lactation, that could affect the lumen to alveolar area and how they kind of proceed forward with their lactation, would be my best understanding based on that.
And when you're doing those measurements, are you like, how many points of the biopsy are you looking at? Are you averaging them, kind of your methods?
So we looked at 5 images per slide, and then we would combine all them all and then take an average, and then we use SAS to look at the statistical results for our results.
Great, thank you.